So small cell lung cancer is characterized by the loss of function mutation in p53 and RB1. And due to the loss of these major tumor suppressor genes, there's an inherent genomic instability in small cell lung cancer. The cell cycle is mainly regulated by two checkpoints, the G1S and the G2M. And due to the loss of p53 and RB1, the G1S checkpoint in small cell lung cancer cells is heavily compromised. And now the cell depends upon the G2M checkpoint for its repair upon DNA damage. There are several master regulators of this G2M cell cycle checkpoint. WEE1 is one of them. So we hypothesized that combining inhibitors to this DNA damage repair target, WEE1 with immunotherapy, would improve or augment the response to immune checkpoint blockade. So in the lab, we treated small cell lung cancer cell lines of tumor-bearing mice with either immunotherapy alone or in combination with WEE1 inhibition. And the result was really remarkable. Very shortly after starting the combination therapy, we saw a dramatic decrease in the tumor volume. In fact, some of the animals had a complete tumor regression, which is really remarkable for an aggressive cancer type like small cell lung cancer. And this was an incredibly encouraging result, which we then found that there is an innate immune pathway called the CGAS-STING pathway through which WEE1 response was actually happening. And that led to making these cancers more visible to the immune system and recruiting more cytotoxic T cells. So our study really provides a preclinical rationale for using WEE1 inhibitor as a therapeutic target for patients with small cell lung cancer. And I think the next step would be obviously to see the response of the combinations in patients with small cell lung cancer, that is to combine WEE1 inhibitor with immunotherapy and seeing whether this drug does as well as it did in our study in the preclinical landscape. I think that's where we're going.